Now the next organelle that we are going to deal with is Golgi apparatus. It is the extension of endomembrane system. You know that in a eukaryotic cell a well defined nucleus is present and its nuclear membrane is present in the form of extensions known as Golgi apparatus and endoplasmic reticulum. Now from your knowledge of endoplasmic reticulum, you must have observed that it is extension of nuclear membrane itself, and it is the site for protein synthesis. Because of the presence of ribosomes on it, the rough endoplasmic reticulum is responsible for the synthesis of proteins. And what about the proteins which are synthesized, where they are going to be packaged and delivered to the cell for other functions? That site happens to be Golgi apparatus. Now let us understand what this Golgi apparatus is all about, how it was discovered, who is the one responsible for its discovery. So the first thing that you need to remember is that Camillo Golgi was the first person who was awarded Nobel Prize for his study and discovering the Golgi apparatus within the cell, and this has helped us in understanding cytology to a great extent, that the protein synthesis which has taken place, which is very vital physiological function of the cell, pardon me, it's not physiological function, metabolic function I would say. The function of protein synthesis and the further packaging could not be completed if Golgi is not present. So we need to understand what the structure is all about, what are the functions and how this structure enables those functions of the Golgi. So first of all, it is, as you can see it in the picture as it is membrane bound. So it is a membranous flattened. Okay, sac-like cisternae are present inside Golgi. So what I am trying to tell you is that these sac-like structures which are in close association with endoplasmic reticulum from this end, please pay attention, endoplasmic reticulum would be present at this end and it would import vesicles towards the cis phase of the Golgi apparatus. Now what is this cis phase? This cis phase is the stacking of the vesicles or the cisternae, not vesicles, it would be the stacking of sac-like cisternae together, and that entire composition would be known as Golgi. Now as you can see, these cisternae, they form sac-like structure. They look like smooth endoplasmic reticulum because smooth endoplasmic reticulum does not have ribosomes and it is also like this only sac-like. It is having a lumen inside it where the proteins are packaged, alright. So the main function of Golgi basically is packaging of the proteins. Moreover, you must be knowing that in the endoplasmic reticulum glycoproteins are being formed. So those glycoproteins would be imported in the form of vesicles to the cis phase of Golgi apparatus, and it would be moving further and further. It would come to the trans phase of Golgi apparatus, which is the maturing phase. From there the vesicles would be formed and they would go further for the usage by the process of exocytosis, and the proteins whichever are being formed, they can be brought into the usage after a proper packaging. So we have a secretory vesicle over here. As the diagram shows, there are cisternae which are having lumen inside them, and few learning points are that in a Golgi apparatus there would be 4 to 8 cisternae in a single stack, and it is a complex network of vesicles and tubules towards the trans phase. Now what is the cis phase and trans phase? Cis phase is the forming phase. It's not phase, it's phase, alright. Forming phase, not the PHASE phase thing, and the trans phase is maturing phase, alright. It is away from the nucleus. Cis phase is towards the nucleus. This maturing phase has a complex network of secretory vesicles and tubules which are present, from where the proteins which are properly packaged are released into the cell so that they can be thrown out or given to the cell, and exocytosis takes place and the vesicle gives out the protein whichever is being formed. And do remember, these membranous outlinings of the organelle are having the same membrane as in the case of plasma membrane. Now the next thing that we need to understand is that how this movement of proteins into the Golgi complex and then it's throwing out in the form of secretory vesicle takes place. Now as you can see in this diagram, there is nuclear envelope, its extension is in the form of endoplasmic reticulum. In the endoplasmic reticulum, protein synthesis has taken place. The secretory vesicles are being thrown into the cis Golgi network, where they are properly packaging. The Golgi stacks, they undergo proper packaging for the proteins which are formed from the endoplasmic endoplasmic reticulum, and from there the transphase takes up that packaged protein. It forms small vesicles, okay. If it is forming late endosome, this vesicle can become a lysosome or it can become a secretory vesicle and come out of the cytosol by the process of endocytosis. So this is all you need to understand about Golgi apparatus, that it is an important organelle responsible for the packaging of the protein and glycoproteins which are formed in the endoplasmic reticulum, and the packaged proteins, they would be secreted out in the form of secretory vesicles so that they can be used in various functions of the cell.